Good morning. Thank you for joining us for Research 411, a talk show focused on sharing best practices and trending research topics delivered by professional staff and prominent UT Dallas researchers from various disciplines. I am Tiffany Willoughby, Research Education Programs and Outreach Manager in the Office of Research. I will moderate today's discussion. Joining me today to discuss the role inflammation plays in breast and prostate cancer and how her research on treatment resistance and alternative therapies makes a hashtag UTD research impact is Dr. Nikki Delk. Dr. Delk is an Associate Professor in Biological Sciences, a Cecil H. Ida Green Professor in Systems Biology Science Fellow, and has been funded by the National Institutes of Health National Cancer Institute. Her lab studies inflammation-induced breast and prostate cancer progression and treatment resistance. After receiving her bachelor's in math from Georgetown University, she was commissioned as an Air Force officer, where she served for 4 years at the Air Force Research Laboratory before obtaining a doctorate in molecular biology from Rice University. She obtained a doctorate to pursue her childhood dream to be a cancer researcher. She was a postdoctoral fellow at MD Anderson Cancer Center and faculty fellow at Rice University before joining the faculty at UT Dallas in 2014. Welcome, Dr. Delk.
Thank you.
Yes, you're welcome. Last month, October, was Breast Cancer Awareness Month, an annual campaign to increase awareness of the disease in which individuals wear the color pink. This month is Movember, an annual event involving the growing of mustaches to raise awareness of men's health issues such as prostate cancer. We're fortunate to have you on the show today to share your breast cancer research and your prostate cancer research. Dr. Delk, can you please tell us how you became interested in cancer research?
Absolutely. So I am heavily influenced by the wonderful women in my family, and my grandmother, who I was incredibly close to, Jean was her name, died from cancer. And actually, while she was going through treatments and actually had to quit her job or leave her job rather because of her cancer diagnosis, she became my babysitter. So we spent a lot of time together. She was a wonderful woman, brilliantly intelligent, very influential, not your typical grandmother. And so it's an honor of her that I became very passionate about being a cancer researcher.
Thank you, Dr. Delk. You currently have a lab at UTD. Can you please tell us what studies are currently underway?
Yes, so we studied both breast and prostate cancer. Reason being is because these two diseases are very similar in their behavior. Both are driven by hormone receptors. So for prostate cancer, it's driven by the androgen receptor. So men will make testosterone that will cause the prostate cancer cells to proliferate and progress. In the case of breast cancer, women or men will make estrogen that will cause the breast cancer cells to progress. And so because these hormone receptors drive both of these types of cancers, and because these hormone receptors function similarly, that's why we study both of these diseases. What we had discovered, we made a really interesting discovery some years back. This was while I was a postdoc and a faculty fellow working in Cindy Farage Carson's lab at Rice University. I made this observation that an inflammatory protein called interleukin-1 had an effect on these hormone receptors. The particular effect that it had on these hormone receptors in breast cancer cells and in prostate cancer cells implied that this inflammation could actually be driving treatment resistance. And so that's the research that I'm doing now here at UT Dallas with my fantastic lab that I have here at UT Dallas. That's what we're investigating, is how inflammation may actually be causing breast and cancer cells, because of the effect on these hormone receptors, maybe causing them to become treatment resistant.
Thank you, Dr. Delk. And how about collaborating with other individuals, either internally or externally, to support your research efforts? Can you tell us about your collaborative efforts and identify any benefits?
So I have to go back to, I mentioned earlier that I was in Cindy Farage Carson's lab as a postdoc and a faculty fellow, a research scientist before coming to UT Dallas. Her philosophy was this kumbaya philosophy. One of the slides that she used to show in her talks was this slide of all these people holding hands around like a globe. And that was her philosophy on research, is that you do it as a community. So she was really big on collaboration. And I sort of picked that up from her, this idea that you don't do research in this sort of silo, very, narrow focus, but that you have to include expertise from other researchers in order to answer your central problem. So with that being said, my background, my training is really in molecular biology. So that's the approach that I take to investigating our problems. But we also do need bioinformaticians. We need bioengineers. We need people that do animal and translational models. We need people that study epigenetics. So there's all these different expertise that need to come into investigating this problem. And so with that, I have been able to build a really nice network of collaborators since I've been here at UT Dallas at several of the UT schools and even outside of the UT system. So for example, we have collaborators at MD Anderson that are helping us with animal models and helping us think about how to translate what we're doing in the lab in vitro or in cell culture using more molecular biology focus, how to translate that into an animal model that would then ultimately potentially be able to influence patients. So we're doing that work with MD Anderson. I'll talk about a little bit more today in terms of some things that we've discovered. We look at genetics, how genes are influenced by inflammatory molecules. And so in order, we get lots and lots and lots of information, and that has to be processed and analyzed. And so that's when that's where bioinformatic groups come in. So they have these algorithms and they can do statistics really well, and they know how to pull out pathway analysis. So we do that in collaboration with UT Southwestern. They have a phenomenal bioinformatics core that we've been working closely with for several years. Then we're also, again, looking in terms of looking at genes, we're starting to get more into epigenetics and chromatin remodeling. And so we're doing collaborations with UNT, University of North Texas is another one of our collaborators. We're looking at biochemistry. That's another avenue that our research is going down. And we do those collaborations and do that work in collaboration with researchers at the University of Texas Health Science Center in San Antonio. And I think I've mentioned everybody. I probably have left some collaborators out, but the point being is that we have expertise all over that we rely on to answer our question. In addition, I talk about collaborators outside of the institution, but we also have collaborations within the institution as well. So with BioE and people in chemistry and mathematics. So and even within the department, I'm in biological sciences. So even within the department, we have people that bring different expertise to solving this central problem of inflammation and cancer.
Thank you, Dr. Delk. I heard you sneak the word statistics in your last answer. Can you tell us and the audience a little bit about statistical outcomes related to cancer patients who catch their cancer early?
Yes, so and specifically related to breast and prostate cancer, if you catch the cancer early, there is a 99% 5-year survival rate. So it's pretty much curable if you catch it early. Part of the reason is because it can be removed. It can be surgically removed. So if you catch the tumor while it's still in the breast, if you catch the tumor while it's still in the prostate, surgery, you undergo surgery, and a surgical oncologist takes the tumor out, you're good to go. Where it becomes a problem is once it starts to spread. That's the problem. And so we call that metastatic cancer. And so once breast cancer or prostate cancer becomes metastatic, the survival rate drops, and it can drop to as low as 30% 5-year survival rate. So that's why we want to be able to emphasize people getting screenings early, as early as possible. And that's why we also want to, as cancer biologists, understand how to mitigate cancer once it has metastasized, because that is not always going to be the case. Everyone's not going to catch it early, and you may end up with metastatic disease. Well, we're here to try to figure out how to treat it so that the survival rate goes up.
Excellent, and we do appreciate your research, so thank you for all of your efforts. Now, are there individuals who are more susceptible to growing breast or prostate cancer?
Yes, so as with other diseases, unfortunately, underrepresented minorities, specifically the African American community, has a poorer prognosis when it comes to breast and prostate cancer. Our incidence of breast and prostate cancer may not necessarily be higher than other groups, but the mortality rate is higher. Part of the reason for that is because of the stage at which the disease is diagnosed tends to be at later stages. The other reason is because of external factors. So access to healthcare, even if you are diagnosed early, are you getting the best treatment? Diet, other comorbidities like diabetes, so other diseases that can exacerbate the cancer, it becomes an issue as well. And then there are some possible biological reasons for it. So there may be mutations that are more prevalent in certain communities or certain racial groups that makes them more susceptible to certain diseases or outcomes.
You were talking about other factors, other health factors, even if somebody has, an autoimmune disease or diabetes, or would you consider those other inflammatory factors that would then contribute or relate to the diagnosis of cancer?
Absolutely. So there's definitely a correlation between disease progression and diabetes, for example, for breast and prostate cancer. So for example, in prostate cancer, there's some evidence that diabetic men have a poor prognosis. So their disease tends to metastasize faster. And so there's definitely the possibility that inflammation can play a role. There's other issues with the diseases outside of inflammation. So for example, diabetes, there's issues with insulin signaling that can affect the cancers. Just to make a point, so there are all these different types of, we call them growth factors and cytokines, that are affecting the tumor. So it's not just limited to inflammatory cytokines like what we study, but there's other molecules as well that your body could be producing that would influence the tumor. And so in diabetes, inflammation is one of those things, but there's other underlying issues with diabetes as well that can affect the cancer. One of the things that we're actually wanting to get into in my lab right now, we're focused specifically on inflammation and its effect on the breast and prostate cancer cells, but we're starting to think a little bit bigger and consider comorbidities like diabetes or obesity and how that would also influence the tumor as well. And so that's one of those collaborations, actually I was talking about earlier, we're going to be doing with a professor in bioengineering and here at UTD to look at that.
Thank you. We have a few questions coming in from our audience, and I'd like to share those with you to answer. With Al Roker's recent prostate cancer diagnosis, he brought up a point that 1 in 9 men will be diagnosed, but for African American men, the number is 1 in 7, and it is more deadly. Does your research address this higher statistic?
So actually, because that's a great point. So the research that we're doing, what we observed happens as a consequence of inflammation on those hormone receptors I was talking about earlier. The influence that it has actually is the types of cancers that happen to affect African Americans more prevalently than other groups. So I'll give you a case in point. Triple negative breast cancer is a subtype of breast cancer that tends to affect younger African American women, and there are no treatments for it. We found that the particular inflammatory protein that we're interested in, that we study, can transform a breast cancer cell that normally has a hormone receptor. It can change it into a triple negative breast cancer subtype. And so what we're going to do now is look at the genes, the gene profile of that cell that's now been transformed into this triple negative, non-treatable type of cancer, and see if we can find another pathway, another way to strategy to kill that cell. That would, in that case, have an impact on women that have triple negative breast cancer. Just so happens Black women happen to be more prevalent in young Black women, happen to have a higher incidence of that subtype of cancer. So in that sense, yes, I didn't necessarily start out saying specifically that my research was going to focus on health disparities. I was interested in breast and prostate cancer in general and having an impact on everyone who is afflicted by these diseases. But it turns out, again, the type of inflammation that we're looking at happens to also have a specific impact on the African American community.
Thank you, Dr. Delk. Next question in the queue: do women who are on hormone replacement therapy have an increased chance to get breast cancer?
If they have the hormone receptor, what we call hormone receptor positive breast cancer, so the hormone replacement therapy will actually can actually activate these estrogen receptors in breast cancer. And so there is some concern about hormone replacement therapy having an effect, but that would really be something that you would want to talk to your doctor about. I don't know that hormone replacement therapy would initiate breast cancer. I don't know that to be the case, but if you had breast cancer and then you wanted to go on hormone replacement therapy, that might be something that you would want to talk to your doctor about, because that could be a contradiction.
Excellent. And another question related to breast cancer: do women with more breast tissue have an increased risk of getting breast cancer?
So I don't know about having more breast tissue, but women, and again, this happens to affect African American women that have denser breast tissue, when you're having a mammogram done, it can miss some spots. And so what is also suggested, if you have dense tissue, breast tissue, in addition to mammograms, is getting an ultrasound done on your breast to try to pick up some of those spots that might be missed by mammograms. So with that being said, it may not be that women with dense breasts, and again, I don't know this, but it may not be that women with dense breasts have a higher incidence of breast cancer. It just may be that it's not caught soon enough to be able to mitigate it, and so then you end up presenting with the disease. But again, ultrasounds are a way that you can do in addition to the mammogram, and you can ask your doctor for that.
I like the additional suggestions that you offered. A few questions related to your research and your research methods, also from the audience. Is your research geared to changing the hormone receptors to increase uptake?
So our research is actually, we want to block hormone receptor activity. So when I was telling you earlier that these breast and prostate cancers are driven by hormone receptors, what that means is that the hormone receptors are actually causing these tumors to grow. So current therapies for breast and prostate cancer are therapies that will stop the activity of these hormone receptors. What happens is, over time, a patient can develop resistance to these therapies that are meant to stop these hormone receptors. Now you have these hormone receptors that don't care about therapy, they're overactive again, now you get the tumor returning. What we observed is that inflammation may cause the cancer cells to be able to function independent of the hormone receptors. That becomes a problem, because if you now have a cancer cell that doesn't care whether it has an active hormone receptor or not, that means that therapy is not going to work. So what we're looking at is, okay, if you now have a cancer cell that can function, whether there's hormones there or not, whether there's an active hormone receptor or not, we need to figure out another strategy, another way to go after that cancer cell and kill it. And so that's why we're looking at what the genetic profile is of cells when they're treated with, when they're exposed to inflammatory cytokines. Because by knowing that genetic profile, we can say, okay, that particular gene went up in these cells when they were exposed to this inflammatory cytokine. They don't care about hormone receptors anymore. This gene W over here went up. Let's go after gene W. Maybe by going after gene W, that will kill the cell. And so that's the idea behind what we're doing.
Thank you, Dr. Delk. Audience members are interested in knowing about your specific research methods. What methods do you use, and do you perform studies on breast and prostate cancer patients?
Okay, so the specific methods that we use human cell lines. So what's been happening over decades and decades and decades is that surgeons have been removing tumors from patients, and then as long as it's IRB approved, these tumors then can be used to create cell lines. And these cell lines are commercially available. We as researchers have access to them, and we can grow them in the lab. So we grow these human cell lines that have come from different people who have been exposed to different treatment therapies. They may have different, racial backgrounds, that sort of thing. So we have these cell lines available to us that we can grow, continue to propagate in the lab in order to do studies. So that's what we're doing in the lab. In terms of patients, so our work is not at the translational level. We do collaborate with people that do translational research. So the translational scientist is going to be the one that's closer to directly influencing patients. Where we would be very interested, or where our work would require or need material, for example, directly from patients, would be once we've identified what we call gene signatures in these cancer cells in response to these inflammatory cytokines, right? Because remember, we're trying to figure out what new pathways to go after. We can take these gene signatures, get panels of breast cancer tissue or prostate cancer tissue from patients that are currently being treated, and see if those genetic signatures are also present in those patient tumors. And then let's say we find out that pathway W, or protein W, is the thing to go after. We can then go back to the physician and say, you know what, these particular patients have that high protein W, and they say no longer need their hormone receptor or no longer dependent on their hormone receptor. So giving them hormone targeted therapy is not going to work. But hey, what if you go after protein W instead? So that's where our work would sort of translate over into the patient world. But we're really interested in looking at the inside, what's happening on the inside of the cell, so that we can figure out what pathway or what protein to go after inside of the cell to kill it.
Excellent. A thorough description, and I appreciate that. Since there are various ways and treatments for cancer, the audience wants to know why you have this specific focus.
So it's a very cool story, actually. So I'll go back to when I was in Cindy Farich-Carson's lab. She has a really cool background. She says she's a bone biologist, and by training. And prostate cancer, when it metastasizes, it primarily metastasizes to bone. So actually, one of the symptoms of advanced prostate cancer is bone pain. So at that point, the prostate cancer has lesions inside of your bone, and it can lead to bone pain. But in any event, she was a bone biologist. And so some researchers at MD Anderson Cancer Center said, hey, can you help us? This goes back to collaboration, right? And bringing people from different expertise. Can you help us to understand why in the world does prostate cancer like the bone so much? We're trying to figure this out. We're trying to understand what's going on in the environment, because prostate cells shouldn't want to live in bone, but these prostate cancer cells do. They get into bone and they thrive. It's not their natural environment. So what's going on? And so when I joined her lab, another really cool story. So I was in Janet Bram's lab when I was a graduate student at Rice, and we studied plants. We studied plant molecular biology. And there was a process that I was studying as a graduate student called autophagy. And some people have heard of autophagy. I think that the Nobel Prize was recently given a couple of years ago for the person who discovered autophagy. But this is a process used by eukaryotic cells, so by us and plants and other different cell types, as a survival mechanism. So I was studying this survival mechanism in plants. When I joined Cindy's lab, I said, Cindy, do you mind if I studied the survival mechanism in this prostate cancer bone relationship? And she was like, sure, go for it. So that was actually how I got interested in that's how I got interested in that particular focus. So it's really trying to look at autophagy and how it might be having some influence on why these prostate cancer cells were loving the bone so much. And so that's really what led to what I'm doing now. So it was one of these sort of, I'm just a very curious scientist. I'm not a man, so I don't have a prostate, so I don't have a particular, so prostate cancer doesn't affect me personally. But I just love science and being in Cindy's lab, that sort of, and in Janet's lab, that sort of led me down the path that I'm at today. And really, because inflammation influences cancer in general, actually, the phenomena that we're seeing in breast and prostate cancer will likely have implications in other cancer types as well.
Thank you, Dr. Delk. Please never stop being curious. I think the world needs more people like you to conduct appropriate research on cancer. What variables are in play when inflammation is a result or a side effect of treatment?
What variables are in play when it's a effect of treatment? So I'm not sure what you mean by variables, but I will pick up on what the question was about treatment causing inflammation. So we do know for some of the prostate cancer treatment, so for example, there is something called androgen deprivation therapy that's done to downregulate the amount of androgen that a prostate cancer patient would be making, so that hormone receptor I was telling you about earlier, so that hormone receptor isn't active. Well, androgen receptor therapy in and of itself can cause inflammation. And so that can lead to, that's one of the things, actually, there's other labs looking at this, other labs that have more advanced animal models to look and see what's happening actually at a physiological level. They see that this androgen deprivation therapy can lead to inflammatory cytokines being produced by the animal, and then that can lead to treatment resistance. But doctors are aware of this, so there are measures being put in place to make sure to sort of think about how we're treating patients, how long the treatment should be. Should it be an intermittent treatment where you do sort of a start-stop treatment to try to mitigate some of these side effects that could happen that could ultimately lead to resistance?
Thank you, Dr. Delk. When inflammation is at play and you have individuals who chronically expose themselves to inflammation, do you have any advice for them? Can you offer any suggestions for holistic practices they can put into play to reduce the chronic inflammation that they're subjecting themselves to?
So there is the proverbial eat healthy, right? Diet and exercise. That's going to be what I think any person would say in terms of trying to reduce your inflammation. You hear about things like antioxidants. So one of the things that inflammation does is causes reactive oxygen species to go up in your body. And so antioxidants is a way to fight that those reactive oxygen species that come as a result of the inflammation. So again, just healthy diet and exercise. And bear in mind that there are a lot of clinical trials looking at some of these, looking to repurpose some therapies that are anti-inflammatory, say for rheumatoid arthritis, seeing if we can repurpose it for cancer, for example. So there are certainly clinical trials going on out there with the intent of looking at some of these anti-inflammatory drugs that we already use as a way to help mitigate some of that inflammation associated with cancer. But in terms of holistic, it's going to be diet and exercise.
Thank you, Dr. Delk. And we do have just a few final questions from the audience before we wrap up today. The first one is, once you find the protein to target, how would you control, destroy, or regulate that protein so as to change or eliminate the cancer?
Yes, so there's a couple of things. There's a couple of things we have to keep in mind. So whatever that protein is, one of the first questions we're going to have to ask ourselves is this protein important for normal cells? Right? Because if it's important for normal cells and our goal is to destroy this protein, that might not be the best protein to go after, right? Because we don't want the side effects of hurting normal cells as well. So we have to look for something that is specific to the cancer cells. Typically, we're looking for things that are overexpressed or overproduced by the cancer cells that a normal cell would not make. So that's number one that we're looking for with this protein W. The other thing too is proteins have different types of functions, and depending on if it's an enzyme, for example, we can target its enzyme activity. If it's a cell surface receptor, we can make an antibody to block its function. So it depends on what protein W ends up being as a protein dictates how we would go after targeting it mechanistically.
Thank you, Dr. Delk. Is it possible for types of prostate and breast cancer to gradually become resistant to the therapy it was previously susceptible to?
Yes, absolutely. That's true. That's where treatment resistance comes in. And I'll tell you, someone said a statement to me recently, and it's a very true statement, is that cancer cells are the epitome of evolution. And so what happens is we give cancer cells a therapy, they're going to attempt to evolve to survive that treatment. And so when we find this protein W, chances are the cells will eventually become resistant to whatever that protein W is. So we're always researching, we're always trying to find the next thing. Okay, we've got protein W, we also need to look for protein X, Y, Z, because we may have to end up going after that as well. So it's going to be this constant research, this constant, chase. And, the thing is, we have so many great cancer researchers out there. We're up to the challenge, and that's what we do.
Thank you. And the final question before we close out the show today is not a question at all. It's a compliment. Your work is fascinating, and we appreciate you being here at UT Dallas. Thank you for taking the time to speak to us today. And yes, Dr. Delk, thank you so much for being a part of our program today.
Thank you.
The audience appreciates all of the valuable information you have shared with us. Interested in attending another Office of Research event? Registration and so much more can be found at research.utdallas.edu.